replication. Okay. Now, one thing that you need to remember is, Crick, Watson and Crick had given something called as the central dogma of molecular biology. Central dogma of molecular biology. So, according to the central dogma, we have DNA. DNA is able to self-replicate it, generate its own kind, self-replication. Okay, so this is called as replication. From DNA, we get RNA. Okay, from DNA, we will get RNA. This process is termed as transcription. From RNA will code for protein. Okay, so this process is called as translation, or otherwise we call it as protein synthesis. Okay, so this is what we are going to follow. So we are now going to do on we are now going to do on the process of replication, or the process of replication. Okay, now see this. Okay, so in replication, what happens? DNA is going to form its own kind. Okay, its own type. It's going to replicate. Why do we use the word replication? Replication refers to the word replica. Okay, that refers to the word replica. Sana, do you know what is a replica?
Yeah, like identical.
Yes, identical, right? Something which is identical. So DNA, it's going to form an identical DNA. Okay, now, so replication. First of all, see, DNA replication was first studied in E. coli bacteria. Okay, it was first studied in E. coli bacteria. Okay, so since we are talking about replication, so we are talking about what replication? We are talking about the prokaryotic replication. This you have to understand. So we know that E. coli or prokaryotic bacteria, they have a double-stranded but circular DNA, and their replication will proceed from both sides. Okay, replication proceeds from both sides. So let us try to understand here. So we have the DNA. Okay, we have the strand of DNA here. Okay, in between the strand of DNA, we do have the nitrogen bases. These are the nitrogen bases. Okay. Now, how do we know that the process of replication, where it will begin? Okay. One more thing, Sana, that you need to understand is replication, entire DNA will be replicated. Okay. It's not a part of DNA is replicated, and replication takes place in part. Entire DNA will be duplicated, but transcription, only a part of the DNA will be used. This is very important difference. Okay, usually we tend to forget, we tend to ignore this particular differences. So now let us see here. Now, how do we know that this particular, strand of DNA here only the process of replication will begin? Okay, so we'll try to understand one thing, that any wherever replication begin, that is that is a specific point. And imagine, suppose for example, this point, this is called as the ori site. Ori site. Okay, or ori, one second. Okay, so when we talk about the ori site, okay, so here ori site means the site of origin of replication. That means replication will begin at this point. So the question is, what is the base sequences of this ori site? So ori site will always be adenine thymine rich. Now it is not guanine cytosine. The reason why, because if it is adenine thymine, we just have to break only 2 bonds. Guanine cytosine means 3 bonds. So adenine thymine rich tells us that okay, much energy will not be required to separate the separate the DNA strand, because between adenine and thymine there are only 2 hydrogen bonds. Okay, Sana, is this point clear?
Yeah.
Okay. Okay. Now see. So adenine thymine rich. So here the enzyme, okay, the first enzyme, it's called as helicase enzyme or unwindase. Helicase will come and attach on the ori site, and what it will do is that it will break the hydrogen bonds. So once it breaks the hydrogen bonds, this is what this is what the DNA would look like. One second. Okay, this is what the DNA would look like. Oops. One second. Okay. All right. So this is how the DNA would look like. Now the DNA has completely unwinded. Okay. So we have here the nitrogen bases. Nitrogen bases here. Okay. And then see, this is exposed. See, the hydrogen bonds are broken. So they are now this is single stranded. Now we know that first of all, the hydrogen bond, second of all, DNA has the tendency to rewind back. Okay, it has a tendency to again form complementary base pair. So we would rather want that, okay, fine, even if it is, if the helicase enzyme is here, I'll write an H so you'll understand helicase enzyme here all. Okay, helicase enzyme. So even if there it is single stranded, what happened, we'll try to make sure that the 2 strands do not rewind back. Okay, now let's see here. Okay, now, so here what will happen is, attached to the single stranded will be special type of proteins. Okay, special type of proteins will attach to the single DNA strand. This protein is called as single stranded binding protein, or SSB, single stranded binding protein. Is this clear?
Yeah.
Okay, single stranded binding. The single stranded binding protein will make sure that the two strands do not rewind. Okay, the single stranded, I'll write here SSB, maintain the single strands. Okay, maintain the single strands. Okay, now what happens here? Now replication will proceed in both direction, from here as well as here. So for replication to take place, now we need two enzyme. We need first of all primase enzyme. Primase will make primer, then we need DNA polymerase 3, okay, or also called as DNA dependent DNA polymerase. That means this enzyme is dependent on DNA only. Okay. Okay. Now see here. Okay. So now what happens? The replication will start to begin. But we always notice that since replication is proceeding in both direction, so in this area and in this area, there are, stress or supercoiling is taking place. What happens when you stretch? What happens? The other side will become, highly stressed because of the uncoiling on this side. This side will experience high coiling, okay, or supercoiling stress. Now what happens if we do not relieve the stress? The DNA will break off. And when DNA break off, what happens? Replication is stopped, and then even mutation can occur. And that will affect the entire functioning of it will affect the entire functioning of the uh, entire functioning of you can say the body. So now we have two enzyme. We have gyrase, which will relieve the stress, and it is found in prokaryotes. And another enzyme, topoisomerase. Topoisomerase, this is found in eukaryotes. But what is their function? Both the function is same, to relieve stress in supercoils. And how they relieve? They will just make a cut little bit. Once they cut, what happened? It recoils back again, okay, in a proper manner. So okay, now so okay, so now the both sides the stress is relieved. So now you can see that due to the unwinding of the two strands by the helicase, and the process of and RNA polymerase proceeding in both direction, you see, observe here a Y-shaped structure. I'll draw with yellow. Observe here a Y-shaped structure is formed. Okay, this Y-shaped structure is called as replication. Replication fork. Okay, it's called as replication fork. Okay, now next. Okay, so now we will see how the process of replication is taking place. What I will do is that I will show only this section, the highlighted section, one section. But you should know that since it is E. coli replication, it is proceeding in both the direction. Okay, so Let's see here. Okay, so what will, all right, okay. Okay, perfect. There we go. Okay, now, so we'll put the two strands here, complementary basis. Okay, now let us uh, name them. Polarity. This is 3 prime, then this will be 5 prime. This is 3 prime and this is 5 prime. So let's see here. So when we are okay, so now what happens, the first, now we know, already know, helicase enzyme will be here. Helicase enzyme has unwinded. Now what happens, the first thing, okay, DNA polymerase is going to read the entire reading frame. Okay, but we should understand from which direction to which direction. Okay, so first of all, okay, first of all, there will be an enzyme locked in this position. Enzyme locked in this position. Similarly, there will be an enzyme locked in this position. Okay, and this enzyme is called as primase enzyme. This is also primase enzyme. Okay, primase enzyme. And what is technically, what is primase enzyme? It is RNA polymerase. Okay, it's RNA polymerase. So RNA polymerase attaches at which end? It attaches at the 3 prime end, but this is the template strand. Okay, template strand, parental strand, template strand. So see, primase attaches at the 3 prime template strand. Rather than putting it here, I'll do one thing. One second. Okay. Okay. So primase. Okay. So primase. So what primase will first do? Let's see. Use a lighter color. Primase, what it will do, it will start adding, okay, it will start adding oligonucleotides. It will start adding nucleotides, RNA nucleotides. Start adding RNA nucleotides. This is 5 prime, 3 prime, no? So here, this is 5 prime. So this will be 3 prime. So the aim of having a RNA, this is a RNA nucleotide. The aim of having an RNA nucleotide is to generate the 3 prime and 5 prime hydroxyl group. Okay. So once, okay, and this RNA nucleotide is about 8 to 9 base long. So once it has reached about 8 to 9 base, what happens? DNA polymerase 3. DNA polymerase 3. What it will do? It will read the reading frame. Okay, it will read the template. It will read the template. Okay, but from which direction? 3 prime to 5 prime, opposite, like this. Once it reads, then only it can add nucleotides. So it will read from 3 prime to 5 prime. Reading frame, please no, please Asana, remember this. Reading frame. I'm referring, read means here, this is a reading frame.
Okay. Are you feeling well?
Yeah, can you hear me?
Yeah, but are you feeling okay?
Yeah, I'm fine. I'm fine. No,
Okay,
no worry. Just a bit of cold.
okay.
Okay, thank you for asking. Okay. So, okay. So, Sana, did you understand this?
Yeah,
Yeah. Okay. So, reading frame. We have a reading frame, right? So, because RNA DNA polymerase has to read. So without reading, DNA polymerase will not know where to add the nucleotides. Okay, where to add the nucleotides. So let's see here. So what happens? One second. Okay, so now once the RNA is added, now DNA, what DNA will do is that it is reading, no? So now DNA will start adding nucleotides. Okay, start adding nucleotides. Now this will go on adding nucleotides. So this is 5 prime. So they will go on adding nucleotides towards the 3 prime. Okay, so here the replication will be 5 prime to 3 prime towards the replication fork. This is very important to understand. Here the replication is 5 prime to 3 prime. See, DNA polymerase reading frame is 3 prime to 5 prime. But when it adds the new strand, that is 5 prime to 3 prime. Okay, 5 prime to 3 prime towards the replication fork. Okay, towards the replication fork. See, Sana, it is going towards the replication fork. Okay, so what happens? This strand, because it is going towards the replication fork, okay, it is what happened here. I'll write it here. Okay, DNA polymerase 3 is able to continuously add continuously add nucleotides. Okay, it is able to, you have to stress on the word continuously adding. Okay, add the nucleotide. Why? Because it is towards the replication fork. So I'll just give an arrow mark to indicate the relationship. Okay, now so this strand, because it is continuously adding, so this strand that we have, this is called as the leading strand. Sana, understood? Sana, understanding?
yeah.
Okay, leading strand. All right, now let's go to the up. Okay, ups. Now here, now what you observe here is that DNA polymerase will read and will go on adding the nucleotide. But you see here, let me write here. I'll write it here. DNA is adding nucleotide. Okay, DNA is adding nucleotide, or rather, first I will not write this DNA. One second. Okay, now see what happens here. The DNA polymerase 3, okay, DNA polymerase 3, what it is doing? DNA polymerase 3 is adding the nucleotide, okay, the addition of nucleotide is from, let me see, 3 prime to 5 prime away from the replication. replication for, see, read this, away from the replication for. Here it was towards the replication for. Since it is away from the replication for, so one new thing is happening here. RNA primer is not able to add the new strand continuously. So after some times, again here primase. Primase. Then we will again have nucleotides and then find DNA. So here the replication is discontinuous. Here the replication, is continuous. Here it is discontinuous. Okay, so this strand is called as the lagging strand. Okay, lagging strand. Did you understand, Sana?
Yeah.
Yes, understood or not? Tell me. Have a look at the picture and try to recall whatever I have given. See, I've given arrow marks everywhere so that you can understand from where to where it is going. Please take a little bit of time, see that. Okay, because once you know thoroughly the replication, transcription, translation is very easy. Okay, you need to understand. Suppose question, very importantly question, yes, why one is leading strand, why one is lagging strand. So the sole answer is leading strand replication is towards the replication for, lagging strand replication is away from the replication for. Did you get it? Okay. Shall I continue?
Yeah.
Yeah. Okay. Okay. Now you see here. Okay. Now let's see here. I'll draw again the same diagram. Okay. So what we had here, primase, here one primase, another primase here, nucleotide, RNA nucleotide, DNA, and DNA. Okay. Now once what happened, now when the entire DNA's replication is complete, okay, entire DNA, full DNA will be replicated. I told you right in the beginning, full DNA, because duplicated DNA has to be formed. No, see Sana, for example, if I have a like for see maybe for example, let's see, yeah, suppose I have a note, I have sort of a notes, okay, here. Now I want to give you this notes, okay, I want to give you this notes. So will I tear the paper and give it to you? No,
No.
what will I do? What will I
Duplicate
do?
it. Copy
Yes,
it.
correct. Duplicated. So that's what. See, will our DNA just tear itself and give? No, right? It will duplicate it. Yes or no? You give a printout. I'll Xerox it. I'll give a print it out. Okay, I'll make some duplicate copies and all, and then I'll give it to you. Same way, our DNA is also duplicating. Okay, so because we cannot break the DNA and say, okay, let's go to one daughter cell, that goes to one daughter cell. No, entire DNA is duplicated. S phase, Sana, you remember with that cell cycle, mitosis we did? Yes, S phase. If you forgot, just recall the notes that I had given you. S phase is where the DNA replication takes place. Okay, now the two strands are completed, but I cannot use this DNA. So now Next, what happens? See, suppose, for example, this is replication is continuing this direction and in this direction. Let me name the give the polarity 3 prime, 5 prime, 3 to 5 prime, 3 prime. Okay, so this will be 5 prime, 3, 3, 5. Okay. Now, as the DNA is adding nucleotide, okay, we will have another enzyme which is called as DNA polymerase 1. What DNA polymerase 1 is doing, it is going back little back. Okay, little back. And then what it is doing, it is reading this last um, formed DNA. Okay, if there is any mistake, it is going to remove this DNA. If there is no mistake, no problem. It will allow the DNA to again add further nucleotides. Again, DNA polymerase. Again, what it will go back 3 prime. Again, it will read. Again, it will, you'll try to check if there is any mutation. So, what is DNA polymerase 1 doing? This work is done by both DNA polymerase 1 and DNA polymerase 2. So this work that both DNA polymerase 1 and 2 are doing, this is called as proofreading. This is called as proofreading. In the exams and all, when question paper is prepared by the teachers and they give you the question paper, what they do, suppose they're making one question paper, they'll make one question paper and 2 to 3 times they'll read. They'll see if there is any mistakes. They will rectify the mistakes. Okay, before that that paper goes for printing and then it is distributed to the student. We are proofreading it. We are making sure there is no mistake, because when you read the question should be read correctly so that you can write the answers also correctly. If there is mistake, your answer will go wrong. Yes or no? So we are making sure that there is no mistake. In the same way, DNA polymerase 1 and DNA polymerase 2 will make sure that when the DNA is formed, there is no mistake. Okay, there is no mistake. Okay, then next, let's see. So, Asana, can you tell me where did, can you tell me, did you understand this one? The proofreading?
Yeah, the DNA proofreading.
Yes, the proofreading. Did you understand? Yeah. Okay, so let's just see. So DNA polymerase 1, it will go a little back and then proofread the entire section. Okay, so if at all there is any mutation, it will remove that section. Okay, so DNA polymerase 1 and DNA polymerase 2 does the proofreading. Now, once the proofreading is completed, we have to remove this nucleotide and primer. So DNA polymerase 1, DNA polymerase 1 second, yeah, will remove this primer. See, it will remove this primer like that. All the primer it will remove, and the nucleotides also it will remove. Now, what we see is that we see the DNA are in fragments. So, again, DNA polymerase 3, what they will do? It will add the nucleotides, DNA nucleotides. But still, our replication is not complete because we still have these gaps. Okay, now see, when the DNA, come to that 1 second. Okay, so once the DNA polymerase 1 removes the primer and all, there is a gap. This gap is called, okay, so what happens, these DNA remain in the form of fragments. These fragments are called as Okazaki fragments. Again, DNA polymerase 3 will add the nucleotide according to the complementary bases. It will read and add the new nucleotide. But still our replication is not complete. We still have gaps here. We still have gaps here. So this gap will be sealed by DNA ligases or ligase. So ligase enzyme seals the gap. So finally, we have the new daughter strand is ready. Okay, then finally, what happened? Enzymes, restriction endonuclease enzyme will make a cut like this, and then it will remove the newly formed DNA, and the other end will join back. And this completes the process of DNA replication. So that's the new daughter strand that will be formed. And then finally at the end, there will be a nick like that, like, they'll be cut and the two strands will separate and the other DNA will again join back. Okay. So that marks the end of replication. Clear? Okay. So next, once the replication is done, okay, now the replication is done. This is the end of replication. So let's see the next, that is the process of transcription. Transcription. In transcription, there is, okay, formation of RNA from DNA. So here, enzyme that will be required is RNA polymerase 3. You can write, if not, no problem. RNA polymerase, also called as DNA dependent RNA polymerase. So let's start with the process of transcription. Okay, so in transcription, as I have said, that RNA formation will take place. So this is in the presence of the enzyme RNA polymerase 3, and the other name of RNA polymerase 3 is DNA dependent RNA polymerase. Okay, DNA dependent RNA polymerase. So when we talk about the process of when we say about the process of transcription, okay, okay, when we say about the process of transcription, we have to know that it's only a section of the DNA that will undergo transcription. Okay, only section of DNA that will undergo the process of transcription. So let's see on this part. Okay, so now how do we know that which section of DNA will undergo transcription? So same thing, DNA, we'll see here. Okay, so now let's see here. So what happens? Not all segment of DNA will undergo transcription. A specific gene is there in DNA that will undergo transcription.
Mhm.
So imagine only this to this section. This is called as transcription unit. Okay, transcription unit. Now, when we talk about transcription unit, so let's see what does transcription unit means. Transcription unit, it is a segment of DNA that will undergo transcription, that will undergo transcription.
Mhm.
Okay. Okay, segment of DNA that will undergo transcription. Now, so it's called transcription unit. So what does the transcription unit even contain? Okay, this is important to know. What does the transcription unit even contain? So let's see here. So what I'll do is that, let me see. Okay. All right, so we'll just fill this, color this so that we can easily see this. Let's see. Okay, transcription unit we have. Okay, so we are going to here shade out the genes. Okay, we are going to shade out the structural genes and those genes which will help us to understand where the process of transcription will begin. Okay, very well. Okay, there we go. All right. So let's see here. So this is a transcription unit. This is a segment of DNA. The segment of DNA consists of this section. Okay, this section here, this section here, and this section here. Okay, so this is called as UTR. Okay, this is also UTR. Now let's see what do we mean by UTR. This is UTR downstream. Downstream. This is UTR upstream. UTR is untranslated region. Okay, these regions will not undergo any kind of any kind of translation or anything. Okay, so this will be a this will be an RNA. In RNA also we will have this UTR. So we'll just consider this as UTR. Okay, or instead of that, let me just have a look. Instead of UTR, let us not write UTR. This will not be appropriate. Let us not write UTR. UTR will go for translation. Okay, we just write upstream and downstream. That will be better. That will be better off. Upstream and downstream. Okay. All right. So we'll write upstream and we'll write downstream. Okay. All right, so upstream and we have here downstream. Okay, upstream and downstream. So here we will have, oops, here we will have, this is the promoter gene, which is upstream. Then we have here operator gene, which is also upstream. And remaining Okay, here we'll have structural gene. And then finally at the end, we will have terminator gene. Okay, terminator gene. So what happens in promoter gene? It is in the promoter that RNA polymerase will attach. Once RNA polymerase attaches to this, it will regulate the operator, and operator will carry out the process of transcription. That means structural gene will carry out transcription. Terminator transcription is terminated. Transcription is terminated. Okay, so this will be the entire segment of DNA where the process of transcription starts. So let us see this in a more detailed manner. Okay, so we will have, this is the segment of DNA. Okay, this is the segment of DNA. Now, same thing, it will unwind, but here helicase will not unwind. Here, RNA polymerase will unwind DNA segment. And which region they'll unwind? Ori will not say ori side, but the segment that they'll unwind is rich in adenine and thymine. Okay, it's rich in adenine and thymine, so it'll be like TATA. Okay, this is called as TATA box or Pribnow box or Hogness box. Pribnow or Hogness box. And it is rich in with thymine adenine. All will have only double hydrogen bonds. So it will, you know what will happen here? It will be easy to break this bonds. Okay, will be easy to break this bond because they have double bond. So here helicase does not play a role. RNA polymerase will unwind the DNA segment. RNA polymerase will do the work of helicase. So just one second. Okay. Okay. So only a segment of DNA we are using. That's it. We are not using entire DNA. Only segment of DNA we are using. Now, this is exposed. So we will have a protein. We will have a protein that I named earlier, SSB, single stranded binding protein. SSB, single stranded binding protein. Single stranded binding protein, SSB. Okay, so
Yeah.
SSB, single stranded binding protein. So let's see here. Okay, no, let's see this. So now what happened? RNA polymerase will be there. Okay, RNA polymerase. So we have here, this is RNA polymerase. This is RNA polymerase. a part of the RNA polymerase contain, okay, a factor. This factor is called as sigma factor. Sigma factor is nothing but initiation factor. So RNA polymerase goes and attaches at this point. Okay, so what does sigma factor do? Sigma factor will first initiate the process of transcription, and also sigma factor will help RNA RNA polymerase to recognize, okay, RNA polymerase to recognize promoter gene. Okay, recognize promoter gene. So now what happens? This RNA polymerase will, what it will do? It will attach to this region, and then then the process of transcription will begin. So gradually we will see that the one new RNA will be formed like this. This is 5 prime and this will be 3 prime. Go on adding. 1, 1, 1, 1 will start go on adding. So this new RNA will be formed. Okay, this newly formed RNA is called as hnRNA. HN stands for heterogeneous RNA or RNA transcript. Newly formed RNA, hnRNA or RNA transcript. Okay, now a time there should be someone to tell that, okay, stop the process of transcription. We have formed enough of RNA. Okay, this long RNA is enough, no need. Okay, so now what happens? There will be a factor which is called as rho factor. Rho factor attaches to the terminator gene. Rho factor will attach to the terminator gene. Rho factor will attach to the terminator gene. Okay, once it attaches to the terminator gene. Okay, once it attaches to the terminator gene. All right, so the process of transcription is terminated. Terminated means stop. At once, what happens? This RNA, mRNA will drop down. This is 5 prime, this is 3 prime. mRNA will drop down, then rho factor removes, and RNA polymerase removes. After that, the DNA recoils back. Okay, DNA will recoil back, and then DNA goes on for replication. So,
Yeah.
what we have here is a newly formed mRNA. This is a newly formed I'll not say mRNA, I'll just say RNA. RNA. And it is also called as hnRNA or RNA transcript. Okay, hnRNA or RNA transcript. Okay, now. Okay, so now, Sana, tell me, can I use this mRNA for translation?
Yeah.
Can I directly use?
No.
If it is prokaryote, I can use. If
Yeah.
it is eukaryote, I cannot use. Okay. Now, if it is prokaryote, directly use. No problem. Prokaryotic genes are not split genes. If this is a eukaryote, then I'll not use. Why? Because will this will contain, this is called as split gene. That means it will contain both coding and non-coding sequence. It will contain both coding and non-coding sequence. Coding sequence will code for protein, non-coding will not code. So now, very important thing, I have to remove this non-coding. Okay. Coding sequences are called as exons. Non-coding sequences are introns. You know, Sana, exons and
Yeah.
introns?
Yeah.
Coding that will code further for amino acid, will code further for protein synthesis, exon. Those which will not code for any amino acid, introns. So now, first of all, this mRNA, this newly formed mRNA is unstable. I need to stabilize it. So, what I will do for stabilization? What I will do in this 5 prime, I will add, okay, in the 5 prime, I will add guanosine residue. And now addition of guanosine residue, this is called as capping. At the 3 prime end, I will add I'll add polyadenylate residue. Polyadenylate residue. That means there will be lot of, like, adenine like that. So I call this as a poly A tail. And addition of polyadenylate residue, this is called as tailing. One is capping, another one is tailing. Capping and tailing. Now when the capping and tailing is done, now this mRNA is stable. Now what I will do is that, okay, suppose this is the exons. Sorry, these are the introns. The red ones are the exons. Okay, so let's see, these are introns. This is exon. Now what happened? I have to remove this intron. So there are enzymes that will come and attach to the introns. Okay, enzymes. These are called as spliceosomes. Spliceosomes, what it will do is that it will remove the introns. So finally, when the introns are removed, this is what we will get.
Yeah.
Okay, we have removed. So it occurs in fragment, but I cannot take this mRNA. So what will happen is that these will be joined by ligase. Now, what happens if you, now this is called as functional mRNA. M stands for messenger. M stands for messenger. mRNA. Okay, now you see the size of the RNA transcript and the messenger RNA. It's very short because we have removed the introns. Okay, we have removed the introns. Now this functional mRNA, it can go for protein synthesis. Protein synthesis. Now, what happened here is that, let's see here. Okay, so we'll go forward. Protein synthesis, this mRNA. Till here, is it clear, Sana?
Yeah.
Yes. This is called as post-transcriptional modification. Okay. It's called as post-transcriptional modification. Post-transcriptional modification, where we modify the mRNA after the process of transcription. Now here, this is mRNA. Here there are only coding sequence. Okay, only coding sequence. Our gene, eukaryotic gene, is a split gene containing coding and non-coding sequences. So we have to remove the non-coding, keep the coding. Till here, is it clear, Sana?
Yeah.
Okay. Now, this RNA, replication, whatever we studied, replication, transcription, all these are taking place inside the nucleus. Okay, inside the nucleus. So let's see now. So once this mRNA comes out, okay, this mRNA comes out, this mRNA will proceed to any ribosome for protein synthesis. Is it clear? Yes, Sana, is it clear?
Hello?
Yes, can you hear me?
Yeah.
Yeah. So is it clear?
Yeah.
Okay. Now mRNA comes out of the nucleus to continue with the process of protein synthesis. So next step is translation or protein synthesis. Translation or what? Protein. Now, when we are talking about protein synthesis, where it will take place? Protein synthesis takes place in all the places where ribosome is present. Wherever you will see ribosomes, there itself you will see protein synthesis. Okay. Let's see a generalized structure. Large subunit. Okay. This is the larger subunit. This is the smaller subunit. There will be a groove. Okay, groove for mRNA. Groove for mRNA. This is mRNA. So, this is larger subunit has three sites: A site, P site, and E site. Okay, A site, amino acyl transferase. P site is peptidyl transferase, and E site is exit site. Okay, E site is exit site. Now, for protein synthesis, we will need another RNA. This is tRNA. T stands for transfer RNA. Okay, tRNA, transfer RNA. Okay, next. So now one important thing that we need to know is that for every amino acid, there is one tRNA. For every amino acid, there is one tRNA. So now the process of translation will begin. Okay, process of translation will begin. Now, the two subunits of ribosome, they are never together. They remain separated. They come close to each other only during protein synthesis. Okay, only during protein synthesis. So the two subunits are kept together. Okay, two subunits are kept together by magnesium ion. It is magnesium ion that will keep the two subunits intact together. Okay, two subunits intact together. Now, peptidyl transferase, P site, peptidyl transferase. Okay, this particular enzyme, it will form peptide bond. It will form what bond? Peptide bond. Very important. If a question is asked, what is the building blocks of proteins? Answer is amino acid. Proteins are made up of amino acid. Okay. So now what happens? The mRNA will enter this groove. mRNA enters this groove. Okay. And here what happened once it enters, now this mRNA will have only coding sequences. Okay, only coding sequences. So what it will do is that the here the RNA, okay, here the RNA or mRNA will first enter into the groove. Once it enters into the groove, it's very important that, okay, a bond is formed. So the first this thing enters, okay, the you can say the first mRNA enters, immediately what happens in the first step, there will be activation of tRNA. The tRNA will be activated so that it can read from the mRNA. So activation of tRNA, amino acylation. Okay, amino acylation also takes place. So Sana, did you understand activation of tRNA?
Yeah.
Without the activation of tRNA, protein synthesis cannot continue. Is this clear?
Yeah.
Yeah. Okay. So Sana, we'll do one thing. We'll stop the session here today.
Okay.
Okay. So we have studied translation. So I don't want to start it today. All right, because
Mm-hmm.
I will be needing an hour or so to explain the process of transcription. Clear?
Okay. Yeah.
All right. Sorry, translation. Okay.
Yeah.
So, Sana, if you have any doubts, anything, you can please inform through the mail. Okay.
Okay.
All right. Okay. So, shall we end the session here?
Yeah.
Okay. All right. Bye.
Thank
Take care.
you. Bye.
Bye.